Thank you for the invitation or allowing me to participate in this workshop. I spent a lot of my time. I'm professor at Concordia University. It's in Montreal, Canada, and spend a lot of my time in sort of messy, theoretical, notational, heavy, worrying about small details world. So it's been, very enlightening to be here and hear about all the different, like the diverse set of applications and biological problems that people have been talking about. So thank you all very much for your participation in this workshop. So the work that I'm going to be talking about is sort of on the more applied side of my work, and actually came out of a working group. So this is a plug for an institute, right? It came out of a working group after a workshop at the American Institute of Math, which is in Pasadena, which is another lovely setup like this, where they allow you to do lots of collaborative work. And it's joint work with some people at UCSD, University of Maryland, and Nottingham in England. So what we looked at is essentially autocatalytic reactions. So the little motivation is that autocatalytic reactions have been sort of gaining a lot of attention in applications that had to do with sort of the origin of life and the figuring out how come the RNA became so complex from such simple things, looking at kind of how do simple networks promote certain kind of complexities in reactions. In all fairness, I don't know much about the specific applications, but I can tell you what autocatalytic reactions on a sort of base mathematical Bayer modeling look like. So essentially you have, you've heard about chemical reactions, so I don't need to do a little intro about that, but essentially you have an autocatalytic reaction cycle, which means that, species in pairs sort of promote each other. And the way they do this, so they promote the generation, regeneration of a species. So you go from having 1A2 to 2A2 in the presence of A1, right? And this kind of happens in a loop. It's not sort of like everybody's promoting everyone, and the loop is something that creates some interesting phenomena. So essentially, you have this like cycle of reactions. And of course, as soon as some part of the cycle breaks, you're sort of stuck, right? Because things can't promote, things can't generate anymore. But then, if you have a production and, degradation to keep things stable, so if you have a production, sort of an inflow of one of these things, then you can keep this running for a while. So the idea is like, the relevance is this, there's a nonlinear feedback through a whole chain of other species, and how big the chain is matters a lot in sort of the dynamics. And there's many applications or sort of various areas of biology where in the Bayer mathematical modeling, these kind of autolytic reactions come up, such as gene regulation, signaling cascades, et cetera. And people have been using it in sort of other non-biochemical reaction setups as well. So I spend my world in, worrying about mathematics, right? Cleaning up the little details. And so what was interesting for me about the system is that it has these like very different dynamical features that can come up. It has the switching, the bistability, some oscillations that come up. And the part that was interesting to me is that stochastic modeling is really important if the species counts are low, and I will argument that soon. So, not living, you can't live in a deterministic world if, you really want to capture what happens when you have small counts, right? And the thing that we were particularly interested in is like, how much sensitivity do you have to these rate parameters? Because in real life, you don't estimate these rate parameters precisely. And the models that we've seen had a lot of like sort of symmetry in terms of the catalytic rates, what they are. And so how sensitive are these models if you perturb the symmetry, right? So if you have unequal rates going through the cycle, even slightly unequal rates. And so you'll see at the end of the talk maybe a bit about like the fact that, the loop cycle kind of structure of this matters a lot, and that the number of species that you have involved, like the length of the chain, matters as well. So the first paper where we saw sort of some development is this paper that appeared in PhysRev E in 2001, where they took a look at this cycle with 4 species. And so this is just a quick schematic so that you see what's going on. Is, the promotion, A1 is helping A2 regenerate, A2 is helping A3, and so on. And there's sort of an inflow and outflow of each of these different species. And so this is one of the simulations that they've given in the paper, where you see that if you're in low count, so V here is, in their notation, is the total number of species. So if you're in low numbers, like 32, as in the simulation, you actually see this, like, what they called discreteness-induced transition, things you don't see at higher volumes, which is that basically rich sort of area, like when you have small numbers of species A2 and A4, then you have large numbers of species A1 and A3. So here in the 4 cycle, they're kind of paired. So if you look at when you have, there are certain things that are very, you can see it better at the end here. At the end of, the end of time, that the red and the blue are very low, when the green and the pink are high. But even between them, right, like you have this switching between which one is sort of more prevalent than the other. So they did some. So again, this is visible, as you'll see in the next one, in one of the follow-up papers when they were doing all sorts of analysis. The simulation that shows, the blue simulation. So this is a 2-species cycle, which is easier to do things in. When you have large counts, 2,000 of stuff. This is the black, right? The black one is, you just have this stochastic oscillations, but they're pretty all stable, like close to some equilibrium value. They're just sort of, it feels like Gaussian fluctuations around some stable value of, here you're just plotting one of them because you have, 2. In this one, actually, they changed the production and degradation into mutation so that they would conserve mass. Whereas if you have smaller counts, like 50, the blue line shows these like transitions between like lots of one and then none and then the other. Okay, so that was sort of motivational. The mathematical framework here is, if you were to do a deterministic model, you had set up these systems of ODEs. Now, in two dimensions, you have like a unique stable point. In multiple dimensions, things get more interesting, obviously. But even in the 2 dimensions, you have more interesting stochastic behavior, as we've shown. And the way we model this is sort of the standard Markov chain model for this. Like you have here, it's mass action kinetics, as Jinsu was talking about, where the rates happen proportional to the number of molecules you have, times some multiplied by some reaction rate, which are these kappas. And then, we're all modeling this in some, density dependent Markov chain based on these reaction rates. Okay. So after these, physics, I don't know if I should call them physics papers. There were some work to try to figure out, like, what's, they did a lot of sort of interesting, trying to figure out what's the mean switching time and things like that. But a lot of that was done based on some approximation, and the approximation invariably hits something like, let's take n, which is the total number of things, to be kind of large, right? Which is not great. But in any case, here, if you can also just look at long-term behavior, right? And so, if you remember the pictures from before, you have these sort of like switching between the states, and you could sort of say, well, if I'm close to the boundary, like, these extreme values always, like, I could look for the proportion of time, that the species spends there, right? Like, so you're kind of looking at it from an ergodic point of view and saying, oh, what's the stationary distribution for the system? How much mass is it at this end of the point? How much mass is it at another end of the point, right? And so there was this paper by Bibona, Jinsu, and Karsten Wüff that said, okay, well, this Markov chain, if you look at the stochastic, the Markov chain is recurrent. It has a unique stationary distribution. with exponentially fast convergence. So this explicit, if you can get an explicit formula for the stationary distribution of the system, then you're approximating the long-term occupation measure of these states, right? So it's giving you some information. However, they could get a precise description of the stationary distribution only in the symmetric case, meaning all the catalytic rates kind of happen, with equal rates. They are exactly the same. All the production degradation. So, you exchange the labels of the particles and you don't know who's who, right? Like it's fully symmetric in that sense. So a student of mine then did some work to try to say, oh, can we extend this to asymmetric rates? Like what if the catalysis is not the same for the different pairs? What if it's slightly higher for some, even marginally, like 1% higher for some of them? What do you get then? And so we noticed some similarity, some analogy with the population genetics model, where they, essentially here, if in the population genetics model, a lot of them live on conserved population sizes, right? So here, if you use production degradation, you're not quite there, but you can make some analogy. And so he did some work to show that whatever the stationary distribution in the Moraine model would be, it matches really well with what it would be in this case, right? So he analyzes like kind of the discrepancy of how far from stationarity it is. Okay, now that's just long-term. One thing that you can notice, this is sort of showing the difference between big N and small n, right? So the first graph is basically, so this is like what the station distribution would look like. And so with large volume, you'd end up with some Gaussian thing around the equilibrium, right? So this is a 2D system, right? So two species, and so you're just plotting what happens to one of the species, and so this one is somewhere at the half point. So this is the symmetric rates that came from the Bibona, Kim, and Vief paper, and there is this phase transition where when n is very small, you're living at these extreme ends, right? When n is big, you are normally distributed around your stable state. And when you're somewhere in between, right, which is like very specifically, you can define it in terms of kappa and lambda, the production rates, the catalytic rates, and the dimension of the system. So that middle one is like the station distribution is kind of uniform on all possible values here. And so it's kind of, it shows like where do you want to live here? And so we wanted to look at, there's these scaling limits, as I said, that give you different values for, different behavior for the stochasticity of the system. And however, this was rigorously done only for the symmetric ones. We showed it, this is the work that my student did wasn't, it's slightly approximate, but he shows that there's again this phase transition, et cetera. Now, there are other results that people have done using a diffusion approximation, but the trick is diffusion approximation. So in these three papers, people considered a diffusion approximation to this, right? But a diffusion approximation makes sense only if n is reasonably large and not if it's small, right? So what we noticed is that basically you can think of it that way, but if you go back to the original paper, you realize that really the reason why the switching happens is that there's a separation of, timescales that's happening here. So essentially, with all these parameters that they did simulations with, it's the catalytic rates that were kind of had an order one rate, and the production degradation were very small, right? And so that's essentially intuitively why you live in these boundaries, is that when you run out of something, you're going to have to wait a bit for it to, for some production to bring that in. And then the catalytic rates very quickly want to equilibrate you to something. And so, or unfortunately, you get absorbed in the other one, right? And then you have to wait a bit more for that, et cetera. And so really what we did is then looked at this multi-scale approach and said, okay, let's just use a different parameter, not the volume of the system. Let's just use a different parameter that separates the time scales of the autocatalytic reactions from the production degradation rates. So that's what you see here when I say, the production degradation of order 1, but, 1 over epsilon is for the catalytic rates. And here we introduced the mutations because we kind of wanted to see how that plays a role, but also there's some technical reason why you have to do it. And that's basically because we're going to use, one of these very nice standard probabilistic tools called stochastic averaging to figure out the whole process in time, how it involves. And so it's clear here because you have production degradation, you're not living on a conserved mass part, but the total mass is actually very slowly moving because the production degradation are slow, right? So the total mass, which is the total volume of your system, is moving slowly. It's moving stochastically, but on a much slower scale. In the meantime, the individual stuff equilibrates to some station distribution conditioned on the value of the current population size, okay? So if you do that, then basically you can write down that the total population size is some birth and death process on this order 1 time scale, whereas the individual species are converging to some stationary distribution that mixes within some epsilon period of time. Okay, but what you need for that, I should say, is that if you want to figure out then how the process behaves, you really need the stationary distribution of the individual populations, right? So the individual species, what do they equilibrate? What do the catalytic rates do in terms of separating them? How much do they separate them into, mass up and mass down? So we analyze now if you go like an explicit expression, if you want everything to be asymmetric and not use some trick, you can really do it cleanly only for 2 species. And you get this, you get some nice clean expressions using these Dirichlet multinomial distributions. And basically it turns out that in the symmetric case, you just get the Dirichlet multinomial distribution, whereas in the asymmetric case, you get what I call, or what's being called the weighted version of that. So these kappa 1 and kappa 2, which are the catalytic rates, are weighting how much you should amplify your symmetric situation. So if you were to set them to be equal to each other, then you would go back to whatever result you would get the Dirichlet multinomial, which is the result that they got in the Bibona, Kim, and Youth paper. So what's interesting now is how does this asymmetry actually play out, in the long time process, but, dynamically in the long time process. So what you need is this mean of each of the species. And if you look at it, the mean of the species, now you go to that explicit expression, you do something with these Gauss functions, and for the case where things are symmetric, it's very easy to compute that the mean of each one is just a proportion of the total number, okay? And so you end up with something that's some fraction for any value of y, right? Whereas in the asymmetric case, you actually end up with something that's constant for large y. So the way you can, it's plotted here. If you plot these explicit expressions, you see that the top is the symmetric case, the bottom is the, nope, that's wrong. Sorry, the top, it has the straight line, the very, I can't tell, is it green? The very linear one up there. And then when you have asymmetry, the curve just starts to bend down and wants to asymptote to something that's constant, right? And the bottom one is, I guess, showing if you start changing the mutations, because you want to see if you get rid of the mutation to eliminate it from the system, what happens. So it's essentially, there's some intuition about why it's no longer linear for large values of y, and mostly it's because the asymmetry is such that you end up spending most of your time, it actually pushes you to spend a lot of the time in one of the states, and so that really skews the mean. And if something is 0, then that's going to not give you a fraction of the total population size. And so what that means for the total population is that now, you had this birth and death process, and you're like, oh, all of the birth-death processes should, converge to something nice. But the station distribution for those is quite different. If you have purely linear birth rates, you get a Poisson limit, for what the overall population, like the total counts, are going to look like. Whereas in the asymmetric case, you get like a linear plus constant or linear minus constant, and then actually that is no longer Poisson. As you can see here, the first one is the symmetric case. It looks kind of nicely tight, the way Poissons are supposed to look like, whereas the second one has a much larger variance, okay? And so it's kind of interesting that you get a completely different total population count as soon as you have asymmetry, plus you get that some of the species end up in constant amounts where some of them are really in the bulk. So we thought that was kind of an interesting feature. That at work, another plug, this time for a journal, that work is actually appearing in the Science Life Sciences journal, which we had a very nice experience dealing with. So to summarize this work, it was sort of, math stuff with tools that are nice, but kind of easy to use, but you can get some clean results out of this. And we get this like all the process level approximation, not just like the stationary distributions, long-term behavior. And we actually observe that, the sensitivity to all of this symmetry that it's nice to assume if you want to do some stuff, actually really shifts the final, dynamics of your process in some substantive way. And one thing that I should mention that we haven't really examined in full enough detail, because you have to figure, we have numerics for larger number of species, but not precise mathematical, analytical expressions for these stationary distributions. But what happens, for instance, in 3D is that you don't have this fragility anymore. And that has something to do with the way that the structure is working out, because you have three things to cycle through. And so you kind of, intuitively, I feel like you can't just get stuck in two of them being low. Whereas in 4 dimensions, when you have 4 species, you again get this feature of like you get really linear of everything symmetric, and then it starts curving down. Anyway, so these are just based on some simulations. And this is sort of like the diagram that summarizes the differences between the symmetric stuff and asymmetric. And you can see how different, these top pictures are about the differences in the 2 species. And you can see how much nicer it is in the symmetric case, whereas in the asymmetric case, it's really living on somewhere where, it's more focused on one of them. And eventually it ends up being able to go to the other one, but in a much more asymmetric way. So I think I'm going to end my talk. Oh, I did have a plug for asking you for interesting examples. So most of my time I spent thinking about some new stochastic processes, which I call measure-valued piecewise deterministic Markov processes. So Jim McLaurin talked about piecewise deterministic process, and so did Maria the other day, right? Like these are processes where you essentially do something deterministic, and then you change some parameters of your flow or whatever. And so here, based again on some motivation from biochemical reactions, we came up with scenarios where you have lots of, because of the difference in abundances in different types of species, you can get to limiting framework where, and the species that are in small counts are actually very important for the dynamics, right? So what they do, whether they're 0, 1, 2, or 3, really significantly alters what's happening with the other things. It's sort of like the gene on off and the proteins around in big quantities. So here our proteins are spatially distributed. So I have a PD that's telling me where the proteins should be, but I have this gene on and off, which is run by a Markovian dynamics, which is going to tell me how I'm interrupting this. And I should mention Sean Lawley worked on these kinds of processes too, with the switching happening on sort of the boundary, whereas here the switching is kind of happening in perturbing kind of the parameters of the flow for this PD of the proteins. So if anyone has, we've established a nice framework for these PDs with these regime switches, right? And also SPDs with the same stuff. So if anyone has, now we're looking at some kind of long-term behavior, large deviations, and all of these things. But if anyone has like some nice concrete examples, it'd be much easier for us to figure out how to do things with them. So I'm grateful for Jim for suggesting some calcium signaling stuff. And if anyone else has some kind of system where this comes up, I'd be super happy to hear about it. Thank you.
Yeah, thank you, Lea. So when the autocatalytic reactions are asymmetry,
yep,
the conditional mean is non-monotonic with respect to the y. When, for example, case 0.9, you show the plot.
Yeah, the mean of the individual ones, yeah. This plot.
Yeah.
Yeah.
It's very unintuitive. For some y, the average of x has been maximized.
Yeah, yeah.
Of course, you have a closed form of the average, maybe, but I just try to intuitively understand why. Why? Yeah.
Yeah, no, and we don't have any sense of like where that maximum is, right? Because the reason is you have this analytical expression.
Yeah.
These are these Gauss hypergeometric functions, which if someone's, you know, really an expert in, they could probably try to figure. But if you try to take derivatives of this, like, yeah, I just, you know,
yeah.
you don't know how to necessarily solve this or figure out. So numerically, you can figure it out, but analytically.
No, just a recursion for the derivatives. You just get more.
Well, it depends. Sure, but it depends here where you take it. There's recursions, but in the different, what do you call it, entries of this of this function in different variables. So there's lots of recursions that are nice that we've exploited, but not in all of them, right? So, yeah.
If I understood correctly, you consider a spatially homogeneous system, right? So how relevant diffusion would be for applications and how difficult would be to implement it into your analysis?
I have not thought about that. That's an interesting problem. I don't, because they need to find each other kind of thing, right? So then that plays into some, you know, slower thing. Yeah. Thank you.
So it seems like you have these bottlenecks where the autocatalytic loop sort of gets stuck because it's a little bit slower, and so stuff like backs up there. So if you have many species though, and let's say like two of them are the slowest, but they're separated or something like that, then do you sort of split the distribution of where you tend to find them and at quasi-stationarity? Like
you could, I mean, I think
like you could put like multiple bottlenecks in a chain.
I think so. We haven't gone down to that level of detail, but I think you can see in these original plots that that's kind of what happens. I think that's what you're talking about.
Yeah.
You're now in a bottleneck at this time, 5750 onwards, you're in a bottleneck of N1 plus N3, and you have more N2 and N4. But then those two, right, like those are also switching between, you know, there's another bottleneck within the bottleneck.
And if you break the symmetry of those, like how, I don't know, can your perturbation theory figure out like different those rates would be?
I think that at this point you could use perturbation theory to do some numerics on it. Yeah.
Oh, okay.
So there are some works that like Ruth Williams at San Diego did for some other, there's a reference to their paper in one of these slides. They did for some epigenetic stuff. And the perturbation is basically in this, well, the problem with this stuff is this absorption, right? And they did sort of, if you think of, if you use a perturbation parameter to sort of, like if you have a little bit of production, like let's say that's an epsilon parameter, right? Like a very slow production, then you get out of the, and if you take that epsilon going down to zero, you have an absorbing Markov chain. But you can figure out what does this pi epsilon go to, right, as epsilon goes to zero. So they have that kind of work in this paper where I think they did it for some epigenetic model.
Yeah, I mean, there's lots of different, I don't know, mechanisms that you can describe for bistable switching in these limits. I mean, the first, I think, is like a
Double well potential.
but
Yeah.
you can also have like
But that's rare events, and these are not rare.
very slowly, right? Which sounds like more of a description of what you have here.
I don't know. I don't know. Yeah, it's not the double well stuff, right? Because that's extreme events taking you from one to another. This is more of a just, polynomial order speed. Like in some sense, the catalytic things are quadratic and the other ones are linear. So the distinction is a little bit different. But yeah, you can, I guess you could describe various things from the Markov chain. So you could figure out hitting times and that kind of stuff, right? Like the switching in that sense, like how long does it take you to switch from one to the other? You can do that.
Does it scale like the hitting time blows up exponentially with epsilon, the time scale separation? Does it scale like algebraic?
I think it's something, well, I have to switch from thinking n, which is the population. So I think it's like log n, like in genetics models. Like fixation time is log in the population size. But I have to rethink whether the n is really the same as the epsilon.
Okay.
It may not be.
All right, let's think Lea again.